My name is Tom Foti, and I'm the president of Aldevron's Protein Services Division. Today, I'm going to be talking to you a little bit about CRISPR products, services, and capabilities, along with our newly launched RMP manufacturing services. So when we think about the market, there's really a range of therapeutic application areas that we're serving. And so the clients that we are working with are doing cell therapy, gene therapy, messenger RNA therapy. They're editing cells using gene editing therapeutic approaches and also vaccines. And so when we think about the products that we need to deliver to enable our clients to have success, those are located in the second column there. So of course, we're providing plasmid DNA for cell and gene therapy, but also for vaccine work. We're making messenger RNA for therapeutic applications now, research grade to GMP, and that's a very exciting recent development. We have GMP Cas9 and SPI5 Cas9 nuclease, and also sell that at research grade. And the thing that really differentiates Aldevron is that we've really tried to focus on manufacturing basically the central dogma of biology, where DNA encodes messenger RNA and that's translated to protein. And so our capabilities from small scale to large scale and from research grade to GMP, so that clients can scale with us, not just in mass, but also in quality, has allowed us to have a differentiating position in the marketplace. So imagine a world where we'd be able to produce curative cell therapies at large scale. And to get to that dream someday, we have to overcome some significant healthcare challenges. Of course, cost is something that's very important to address early on. And also quality is very important. And so the solution just might be allogenic therapeutic strategies that allow for the engineering of curative cells. And you could develop these curative cells by using gene editing techniques or an RMP, which would knock out certain genes and then knock in other genes that would be specific for the cancer that you want to address. And if you could make those edited cell populations and then be able to expand them with quality, you would be able to reduce the costs. And hopefully if you could reduce the cost, you could serve larger and larger patient populations. So in 2017, Aldevron took a bet on launching a GMP off-the-shelf solution, and we were producing the products at research grade, but the idea of producing off-the-shelf GMP was a novel thing in the industry at the time. And so since then, we have produced several batches, both at research grade and at GMP. And we currently offer the products on the left at research grade, and on the right, we have products that are offered at GMP and GMP source grades. And so we have the 1 mg and the 10 mg sizes in all 4 formats. So the whole idea here, just like I mentioned on the previous slide, is to be able to offer a research grade solution. And then when you get in maybe to phase 1, you can use GMP source. And as you move to either pivotal or commercial grade, you have a GMP solution, all with the same vendor. So I want to talk to you a little bit about RMPs, and our experience in producing the enzyme naturally led us to conversations with our clients. And those conversations often resulted in questions like, hey, could Aldevron make an RMP complex at GMP? So we would have a single reagent that would move into a gene editing event. And so here in the slide, you can see in green is depicted our Cas9 protein, and the guide RNA is complex then with the Cas9 protein to make the RMP. And so if we were going to kind of move toward a place where we could solve this problem for clients and have GMP RMPs, one of the first things we needed to do is to develop a testing panel so that we could both look at the functional performance at the date of manufacturing and do product release, but we could also, from a stability study standpoint, test products over a period of time in a way that will give us qualitative and quantitative information so that we could support our clients who are filing IND, IND documentation. And so here we highlight the concentration assay and the residual free guide RNA and residual free Cas9 assays that were developed. So now I'm going to talk to you a little bit more about those. So one of the things we had to do was to optimize our RNP complex, and to do that, we had to develop a way to measure the functional performance. So we created a platformable assay to optimize RNP complexing, determine the linear range for cutting, and to determine the limit of detection. And I say platformable because every time we manufacture RNPs, we might be using a different enzyme complexing for sure with a new guide. And so we wanted to be able to functionally test that. And on the left, you can see our RNP complex titrated. And you can see that on the far left, it's the highest concentration where there's the most cutting. You can see that in the conversion of the linear plasmid DNA with the cut site in converting into two cut bands. And we also developed an in vitro activity assay that was plate-based, that's very sensitive. And again, this was helpful for us so we could determine the linear range of the assay in the EC50, which is the enzyme concentration that gives half the maximal response. The other thing that we wanted to develop was to optimize the guide RNA Cas9 ratio for the complex. And so it's important that the Cas9 was complex, but we wanted to minimize the extra free guide in solution if possible by developing an assay to do that. And so first of all, in this assay on the left, we're titrating the amount of guide RNA. And there is a correlation between the amount of guide that's added and the amount of cutting that you see. And so from left to right, you'll see that the bands on the right have a higher conversion of the linear template to the two cut bands. And so this formed the basis for us to move into more robust testing. On this slide, we're measuring free Cas9, and we developed an HPLC-based chromatography assay to be able to do this so that we could distinguish between RNP and free Cas9. So on left in figure A is Cas9 protein alone. And as you can see in the chromatogram, it elutes off at between 15 and 18 minutes. And on the center panel, we have RNP, and the RNP elutes off between 9 and 13 minutes. And so we could get that resolution between those two molecules, which was very important. And the reason it elutes off earlier is that you have the negative charge from the guide that's on the RNP complex. And so when we overlaid on the right the two chromatograms, you can see the difference in the resolution in HPLC, and you can see that separation, which is important as we started to develop the tests. One of the things we found out that's super important when you're looking at the impact of quality is it starts with high quality reagents. And it's very important that your guide RNA is purified with HPLC. And in our assays here below, it shows why. On the left, you can see a gel where lane 1 is HPLC purified, and lanes 2 through 6 are not. And you can see the smearing above and below the target. And we see the same thing when we do HPLC on the right, where the high purity guide can be resolved, but the lower purity guide has this big shoulder that creates a problem. The next thing we wanted to do is measure free guide RNA. And after evaluating several methods, we used this native page gel electrophoresis to find out and became basically a reliable indicator of unbound guide. And so we ran 2 identical native page gels. The left one is is just a stain gel, and on the right one, it is transferred to nitrocellulose so that we could perform a Western blot. On the left, it's indicated by the arrow. You can see the lower band indicating the guide RNA, and the reason the intensity increases, the concentration increases from left to right. After transferring to nitrocellulose, we probed the Western blot with an anti-Cas9 antibody, and you can see that the antibody lights up on the Western on the top half where you have the complex molecule, but below where the free guide is in excess, it's not detecting it. So that became a helpful assay for us to determine the free guide RNA. One of the most important things that we needed very quantitative assays for that were very robust is we wanted to run a long-term stability study. And you can imagine that when you're going from research grade to tox studies to phase 1 clinical to your pivotal trial and maybe to commercial, it's a long period of time. And the client needs to have the assurance that the product is going to be stable and perform at a high level and not break down over that period of time. When we looked into the data, we found that there was little data out there to support long-term storage of RMPs. And if the goal is to qualify and bank batches of these off-the-shelf products, we needed to understand this critical parameter for the product. So what we did was we developed a stability study where we looked at RMPs stored at -24, -30, up to 6 months, and at 80 degrees, we went all the way to 36 months. So here's a little bit of the data to share. And so this is the 6-month time point, and this is our activity assay that previously I mentioned to you. And as I mentioned earlier, the RMP is titrated from left to right at each one of these different time points. And so on the far left is the activity assay from the release after the date of manufacture. And then it's -80, -24 degrees, and 30 degrees on the far right. So if you can point your attention to the downward yellow arrow, you can see that at -80 and -20, that the enzyme is cutting and converting the linearized plasmid with the target site to the 2 cut bands. It tails off maybe a little bit at 4 degrees, but we see a more significant tail off at 30 degrees, which you would expect. And so that was helpful information and kind of demonstrating to us that our assay was working. So we did the HPLC assay, and so this is data from that. And so these are results from 6 months looking at stability at the different temperatures. And so on the far left is -80, and on the bottom right is 30 degrees. So you can see what the significant difference is between the peak that you see at 80 degrees, and you see maybe a little degradation at -20 and 4, but you see complete degradation at 30 degrees. So this was good data demonstrating that the -80 stored material was stable. This is looking at free guide, and so again, we're increasing the concentration from right to left, where the highest concentrated is on the left. And then we are looking at -80, -24 degree, and 30 degrees once again. And so this is showing at the 3-month on the left and the 6-month on the right. And we were able to see again at -86 months that it was still intact. So we're happy to see that information. Moving to functional performance, this is results at 18 months. All right, and on the left is 6 months at -80, and at the right is 18 months. And so this is again showing that you're seeing the conversion from the linearized plasma to the 2 cut bands, and so that was good to see that we could prove that both by HPLC, which you can see on the bottom, but also as functional performance. And so with that, that concludes our talk today. But I'd be remiss if I didn't acknowledge our great team at Aldevron. And in this picture is colleagues both from Madison, but also colleagues from our Fargo site. And we had a great 10-year anniversary party. So that's what this was from. You can always contact us online through our online submission form on our website, or you can always contact us at protein@aldevron.com. Thank you very much for your attention and have a great day.